Welcome, everyone, to Downeast Institute's third climate change seminar of 2022. We want to thank you for joining us. My name is Sarah Randall. I'm the Associate Director at Downeast Institute, and this seminar is part of our series of Downeast Discussions, where we share some of the groundbreaking research being conducted through Downeast Institute at our facility in Beals and also in the field across the state. This summer, we're offering these seminars primarily every Tuesday during the lunch hour from now until August 16th. And these seminars primarily focus on climate change impacts to commercially important shellfish, which is what we specialize at Downeast Institute. And for those of you that don't know, Downeast Institute is a nonprofit marine research laboratory, marine science education center, and shellfish production hatchery located in Beals, Maine. And we're in Washington County in the heart of what is called the Downeast region. So we also serve as the marine science field station for the University of Maine at Machias. And today we have an hour to talk about today's topic. And our seminar will last about 45 minutes, we believe. We have two presenters. And we're scheduled to end at 1 p.m. And so this is a webinar. I mean, it's not a webinar. This is like basically a general Zoom meeting. So we ask that everyone keep themselves muted during the presentation. And after the presentation is over, we are going to take as many questions as possible, keeping in mind the 1 p.m. end time. And you can ask a question by raising your hand on Zoom or by adding it to the chat. And this presentation is being recorded and may be uploaded or will be uploaded to YouTube. And I would also like to thank Hannah Green, who is Downeast Institute's Island Institute Fellow, and she is helping do the help with moderation and technology today. So I would like to thank her for everything that she is doing today. And we're really excited to be able to share this work with you today on this seminar. We're going to hear from Dr. Brian Beal and Dr. Robert Holmberg. And we're going to be talking about how, different efforts to use shellhash for buffering mudflat acidification, specifically talking about like how the different crushing of the shells and their sizes and their density impacts commercial bivalve recruitment, sediment pore water chemistry, carbonate chemistry. So Dr. Brian Beal is regarded as one of the world's top experts on softshell clams. He is a professor of marine ecology and a director of the Marine Science Field Station for the University of Maine at Machias. And he's a native of Jonesport and has a PhD in marine bioresources from the University of Maine at Orono. And Dr. Robert Holmberg came to Downeast Institute from UMass Boston in 2019 as a postdoc tasked with designing and building an ocean acidification laboratory, which he has done and now is his laboratory here at Downeast Institute. And he's a research scientist here, and he studies ocean acidification and warming impacts on larval bivalves. And he also dabbles in carbonate chemistry monitoring on Maine mudflats, which he'll be talking about in next week's seminar. And with that introduction, I would like to hand it over to Dr. Brian Beal, who is going to give an overview of sediment buffering studies that we did in the mid-2000s, and then our big study that we did last year in 2021 using recycled oyster shells. So thank you, Brian.
Okay, well, thanks, Sarah. So I'm going to be talking about two different studies. As Sarah said, one of the studies was done in 2014, 2015 in the town of Freeport, and the other one was done last year, 2021 in South Portland. So let me see if I can advance the slides, and there we go. All right, so part of the whole reason for my involvement in this sediment buffering story is staring you in the face, which is there's a problem with softshell clams in terms of landings. You can see that since around the mid-1970s, landings have declined by nearly 75%. What most of our data indicates is that it's not because clammers are getting tired of clamming, it's because clammers can't find a lot of clams that are out there. So there's fewer clammers certainly, but there's fewer clams as well. In fact, it's very difficult to find commercial quantities of clams near the low intertidal and the mid intertidal. So if you did a drone survey of where clammers are plying their trade right now, it would be in the upper intertidal or the upper parts of estuaries throughout the state. At the same time that clams have been declining, seawater temperatures have been increasing. And what you see here is a graph that I put together from data that was part of the ongoing data collection that is happening at West Boothbay Harbor that's actually occurred since early in the 1900s. I think it started around 1905 or 1906, but this is looking at simply wintertime temperatures. So each one of the dots is an annual mean for the period between the 1st of January and the 31st of March for every year from 1945 to 2018. And as you can see, there's really been two incidents of real warming, one that occurred beginning around 1950 and going through until about 1955, and then the other one, which has been a gradual increase of seawater temperature over about 40 years that has happened since the mid-1980s. Recently, Andy Pershing at GMRI published a study looking at cod landings and sea surface temperatures and noted that between 2004 and 2014, the Gulf of Maine was warming at a rate that was greater than 99% of the global ocean. So we have these two scenarios. And when I started working in Freeport area, basically the headlines were reading that our flats are acidic and that's really a cause for the decline in the clam population. The pressing problem was the loss of clams, and the studies that linked ocean acidification and sediment poor water chemistry to clams was done in the lab and also in the field. A colleague from the St. Joseph's College, Mark Green, did some studies looking at Mercenaria, the hard clam, in the lab and showed death by dissolution, which was part of the title of Mark's paper in Limnology and Oceanography back in 2009. And then in 2013, he conducted a study in the field at a flat in South Portland that ultimately we used in 2021 that showed that at least over the course of the first 35 days of softshell clam's life, if you buffer sediments on flats, what he showed was a twofold increase in terms of clam recruitment compared to unbuffered controls. Friends at Casco Bay had done some studies and interviewed clammers around the Casco Bay area and found that flats that were no longer productive typically had lower pHs than those that were deemed productive. So we had all this work that was being done, as well as some legislative work that a commission occurred in 2014 and 2015 while we were starting some of our studies in Freeport. And that commission, I'll only read this last one here. You can read them all if you have the time, but studies have also shown, this is in the report, that some bivalve larvae and small juveniles exposed to acidified conditions are smaller. They're less fit, they're slower to develop, and they show significantly greater mortality than larvae exposed to ambient conditions. So the acidity of the sediments can also influence the settlement of these bivalve larvae, certainly for softshell clams. And in more acidic sediments, it's more likely that the larvae will simply encounter that bottom, swim back up into the water column, rather than settle in that acidic mud. But when they do settle, in more acidic muds, they have significantly higher mortalities. So all of this data suggested that and led to a recommendation by that legislative commission to spread shells or other forms of calcium carbonate in bivalve areas to remediate the impacts of local acidification. So we did just that. But we added a twist to it, and that was based on studies that I've been doing up and down the coast for about the last 30 years, where I've found that predation seems to be an important factor in regulating clam populations. So what's the difference between predation and what you might get if you wanted to do what has been recommended, which is simply put shells out in the ocean or on the mudflats to see if that will enhance or increase the pH and actually buffer those sediments. So what we did was we set up a field experiment, and we repeated the same study in 2015. We started at one site in Freeport in 2014, but the experimental design was similar in both years. We used 3 different levels of crushed softshell clam shells, and these were simply shells that we got from a pile of weathered shells here on Beal's Island. We put those clam shells in a fish tote, and we just simply walked on them, kind of like making, wine from grapes. And the pieces of shell would have varied in size from the size of a dime to probably about the size of a quarter. We used no shells in some plots, and then we put 13 pounds of this crushed shell. Why 13? Don't ask, it's just a number. And then we doubled it. Again, who knew what we were going to find? So none 13 and 26 pounds in 10-foot by 10-foot plots. And then we had 2 levels of predator protection, either none, or we used polyethylene netting with a 4.2 millimeter aperture. And every level of factor A was combined with every level of factor B to give us a factorial design of 6 treatments. So this is what our schematic would look like. There's about 20 feet between each plot in rows and columns. And so here are the 3 treatments that do not have nets over them, no crushed in 13 and 26. And then here's the treatments that have been netted. And there's 3 there, obviously 0, 13, and 26. So there's 5 replicates of each of these treatments in this plot. And this was a study that was put out in the field in early April or May, depending upon the year, but prior to clam spawning. And we left it and went back and then destructively sampled using core samples. So we used a 2-pound coffee can core or a number 10 can. We took 5 cores in each one of those 30 plots. So we had 150 samples to sieve, and we used a 1 millimeter sieve to wash the sediments through. We first started by going out with folks from the Friends of Casco Bay that had pH meters that were, and we asked them to find flats that were highly acidic. And so early on, we looked at these 5 flats that were in the Freeport area, and the one that had the lowest was Staples Cove, and that's where we set up this study. So there's where Staples Cove is, and this is 2014, and here's some shells going into a control plot. Here's a control plot that doesn't have shell but has nets. This is an open plot that has, I think that's 13 pounds of crushed shell. There's one that has, it's netted and it's 13 pounds of crushed shell. So there's just some quick shots of some of those treatments. And this is what we found for results after, again, April to October timeframe. So this goes prior to spawning and then well after settlement has occurred. And when we say Maya or softshell clams, we're talking about early recruits. These clams are typically less than 10 millimeters in size. And we found no effect of either netting or shell in terms of softshell clams. In 2014, we also found another species of commercial bivalve, the hard clam, Mercenaria. This was the only year that we found this particular species. But in this case, there wasn't an effect due to shell, but there was an effect due to netting, and we found about 40 times more animals in the plots that had nets on them than those that didn't have the nets. The following year, we did the same thing, except we wanted to set this up in 2 different spots. So we had 2 sites, both of these, again, following the way that we did it the first year, showed that these were two sites that had very, very low, less than 7.5 pHs. And what we found, this is now on the y-axis, I'm looking at pH. We went back in October at the time that we were sampling the experiments or taking the cores. And under the nets, we found that there was a significant difference in pH. There was lower pH. So if you will, the conditions under the nets showed a higher level of acidity. And what we found for softshell clams, remembering that we didn't find any Mercenaria in year 2. But again, we found an enhancement due to netting, but not to the presence or absence of shell. And that was about 70 times more in the nets than the unnetted. And this is also occurring at the other site as well. We're getting 23 times more at the second site. And that study has been published, and it's in something called the Journal of Experimental Marine Biology and Ecology. It came out in 2020. And so to summarize some of the things that we found in that particular series of experiments, we were unable to show any kind of a statistically significant effect due to the presence of shell. So we didn't find that association between acidic mud and softshell clam production. We also did at the same time, and I don't have time to report on them here, but in the paper you can read about the small scale studies that we did, where we actually used a variety of different kinds of crushed shell, and whether or not that was a limestone or an Australuridae or softshell clams. But we also found the same sorts of results, which was we didn't really find anything that was significant that had anything to do with shells. And if we found clams, they were basically in the treatments that had nets on them versus those that were controls. Clams recruited more heavily in those large scale netted plots in 2 of those 3 trials, and in 3 of the 5 small scale trials that we did, even when the pH was, again, lower in those netted plots than in the controls. And it also was interesting that softshell clams were larger in the netted plots than they were the controls, simply, we think, because the netted plots kept predators at bay somewhat, certainly better than having no net at all. And so the predators were simply eating those softshell clams, and they weren't able to eat as many in the netted plots. And so we found larger clams in those netted plots. And there's the distribution of sizes of the control of the open units on the top, and the size distribution of those that were in the netted units on the bottom. Okay, so all of that leads up to where we are today, which is talking about the study that Rob will report on in terms of carbonate chemistry, and I'm going to report on in terms of recruitment of softshell clams and hard clams and green crabs. So we got together with some folks from the Casco Bay Estuaries Project, Curtis Boland, folks at the DMR, Teresa Torrent, and at the US EPA lab in Boston, Matt Liebman and Dave Morgan, and Carolina Batistas from MIT. And basically, we got together and said, well, okay, you use lots and lots of crushed softshell clams. Someone has been collecting oyster shells from restaurants because, you know, they'd like to see field studies with crushed Crassostrea virginica. All right, that's great. So how about using eastern oyster shells as crushed ones? So we did that. And we decided that we would vary the size of those crushed shells, because I didn't pay any attention to that in the earlier study. So we used a small, medium, and large, and I'll give you a size frequency on those in just a sec. And then we wanted also to vary the density of crushed shells per unit area. And our lowest level of crushed shell was the highest level that we had used in the previous one. So our low is essentially equivalent to that 26 pounds per 100 square feet, and I'll show that in just a minute. And then I wasn't going to do a study unless we were going to exclude predators from some of the plots and not others. So that's what we have. So we went from Freeport to South Portland, and that's the site called Mill Cove, and that's actually the study site where Mark Green had set up his study in his publication in Estuaries and Coasts in 2013. There's our oysters, and they were weathered for a year, coming out of probably Casco Bay and going into restaurants in Portland, but they were out of the water and shucked and just dead shells for a year. This is the sizes that we were ultimately able to create. the large, the medium, and the small, 9.4, 5.6, 3.9. These were added to 1 meter square plots on this flat in South Portland near the mid-intertidal. And again, I said our low, I'm not using a number per square feet anymore. I'm sorry about that, but that 1.27 is equivalent to 26 pounds from the previous study. And then essentially we doubled it to 2.54, which would have been what, 26 times 2, 52 pounds per 52 pounds of shell per 100 square feet, and then halfway in between is about 39 or 1.9 kilograms per square meter. This is our experimental design. We used, again, 3 levels of shell, 3 levels of density, 2 levels of netting protection. So that's 18 treatments. And we also included 2 control treatments, which were plots that were where we had no shells and no nets, and then plots where we had net but no shell. So that's actually 20 treatments. We replicated each treatment 6 times, and we took 5 cores for each one. So in this case, we had 600 cores, core samples. Again, those were all washed through a 1 millimeter sieve. This is just some photos of how we installed the nets around these plots. This is just that 4.2 millimeter polyethylene flexible netting. And here's a picture of that netting. We used a styrofoam float as we had in the previous study to try to lift those nets off the flat so that if any clams did settle, that netting wouldn't interfere with feeding, etc. And so there's a kind of a Beal's eye view of the plot. And here's Rob and I at the end of the getting it established back on, I think it was April 22nd, down in South Portland. In addition to that, we also put out something called recruitment boxes, because if we found that there wasn't any clams in any of the cores or few clams at all, is it because the clams just simply weren't settling on the flats, or was it because they settled but then something happened? So we put these recruitment boxes out, and the recruitment box is a 1 foot by 2 foot box. The material is made out of strapping. There's nothing in the box. There's a PET screen top that's 0.9 by 1.7 millimeter mesh on the top, and there's a woven landscape fabric on the bottom. These are deployed on the mudflat surface, and they're anchored with laths at both ends that are knocked into the mud about 20 inches. And then trap nails are pounded from the lath into the side of the box, and it anchors those boxes just nicely. We put 20 inside the matrix, the experimental matrix, and 20 on the outside, just if there had been a matrix effect, we might have seen it. And why we do this was to essentially provide evidence that there was actually a pool of clam larvae and post larvae, at least in the water column. This is Sarah putting in one of the recruitment boxes on the flats. And what we found, and what I could report on, but I won't, a subset of these, but we did find Maya. We found razor clams, ensis. We found blue mussels. We found Atlantic surf clams, spatula, and we certainly found Carcinus. What I didn't show you there was that we also found the hard clam, Mercenaria. This is what one of the recruitment boxes would have looked like. And you can see those holes, some of which come from polychaete worms like sandworms, etc. And so here's the results of the boxes. And we didn't find a lot of clams, but we did find, on average, about 85 clams per square meter. We found 18 mussels per square meter, 20 razor clams per square meter, 25 Atlantic surf clams, and about the same number of green crabs as we found of softshell clams. And in all of those cases, the averages that we found inside the matrix versus outside were not significantly different from each other. This is the size frequency distribution of those clams from those recruitment boxes. And I'm almost done, Rob. There wasn't any difference in terms of the size frequency between inside versus outside. So here's a combined look at the sizes. And I want you to note that we found clams in these boxes as great as 29 millimeters with an average of around 14 millimeters in size. So When we combined all the results from the boxes and looked at the size frequency distribution of green crabs, we found plenty of green crabs, again, at the same densities as softshell clams. And they also had a very similar size range from about 2 millimeters to almost 30 millimeters in size. So what did we find? Maya arenaria in plots that weren't netted. We found of 300 cores, only 6 of them contained softshell clams, and there wasn't an effect due to any of the treatments. In the netted plots, we found even fewer softshell clams and no effect due to the shell. For Mercenaria, we found essentially the same thing. Very few and no effect of the treatments in terms of shell, but also no effect due to the presence of netting. The last slide that I'll show is what we found with green crabs. And this might also explain why we didn't find many bivalves in the netted plots. Here are what we found for green crabs in those unnetted. And here's what we found in the netted plots, which were essentially plenty of small green crabs. So there wasn't an effect due to shell, but there was a statistical effect, significant effect due to the presence of the netting that enhanced the presence of green crabs. So I'm going to stop right there. There's our results. It suggests basically the same sorts of things that we found in Freeport, which was there really doesn't really make much sense to put crushed shell out on the flats. It's not really going to result in an enhancement of softshell clams. So I will stop the share and welcome Rob to the fold. Hey, Rob.
Hello, I will share my screen. Can everyone see that?
Yes.
Okay, great. So I'm Rob, and I'm going to talk to you about my role on the same project that Brian spoke about, and specifically the 2021 project. And my role was analyzing any impact on pore water carbonate chemistry by the shellhash. All right, so just I'd like to briefly go over ocean acidification. Atmospheric CO2 levels are on the rise and have been for decades, due mostly to human activity and especially fossil fuel combustion. That's the red line. This is resulting in a warming of the atmosphere in the process we call global warming. This is why, for example, the United Kingdom is currently on fire. It's also warming the surface ocean in the process we call ocean warming. This is why American lobsters are moving north out of Rhode Island and Massachusetts. But at the same time, a good deal of that surplus atmospheric CO2 is dissolving in the surface ocean, where it's shifting seawater carbonate chemistry equilibria in such a way that pH and the saturation state of aragonite are reduced. And these changes are happening lightning fast in terms of geological time. And they're happening so fast that they're likely to outpace the rate of adaptation in a number of marine species. Right, so when I talk about carbonate chemistry, I'm really talking about 5 different parameters, of which pH is one. And the parameters that are most relevant to the health of marine species, and especially calcifiers, are pH and the saturation state of aragonite, or omega. And omega is simply a measure of the abundance of calcium carbonate minerals dissolved in seawater. It's measured on a scale of 0 to 5-ish. And marine calcifiers like it when omega is really high because it means that there's plenty of calcium carbonate with which to build healthy, strong shells. They don't like it when it's low. And so that's why it's a problem that OA is pushing it lower than normal. But it isn't really possible to measure omega directly. However, luckily you only need to measure 2 of the other parameters to calculate the remaining 3 parameters. And so what I do in my lab is I generally measure seawater pH and seawater alkalinity, and then calculate omega and the other parameters from those. All right, and so on the left are the chemical equilibria that govern the carbonate chemistry system in seawater. And those same equilibria apply to pore water, that is the water in between grains of sediment in the mud on mudflats. However, pore water tends to have lower pH and omega than the overlying seawater. And this is because the mud is full of organic matter. So all the dead stuff and all the poop sinks to the bottom, where it gets broken down by microbes who literally convert the carbon from the organic matter into CO2 that they exhale, which then reduces pH and omega in the pore water. And so although, we expect ocean acidification to reduce the pH and the omega in seawater, we expect it to have the same effect on the already lower pore water, such that it's being pushed below its natural minima. And this could be a problem for things like clams and other species that live in the mud. All right, and I'd like to say a few words about the method that I'm using to measure pore water alkalinity, because it's a method that I put a lot of effort into adapting from the literature specifically for this project. And so I went with the spectrophotometric total alkalinity titration method developed by Bob Byrne and Sherwood Liu for its very high accuracy and precision. However, I very quickly found that those authors have not made their custom software for guiding the titrations available, and also that I had the wrong kind of spectrometer for doing those titrations. So I adapted their method to my spectrometer, firstly by developing my own software, which wasn't easy because I'm not a programmer, one, and two, I had to use a language called ADL to program the spec, and ADL is based on this like ancient implementation of BASIC called SACS BASIC, which no one has used since the 1990s. So that took some figuring out. I also worked with DEI's own Jeff Balicki to develop a custom cell holder, which mounts inside the spectrophotometer. And what it does is it holds the optical cell containing your sample over a magnetic stir plate to keep it stirred, and it aligns it with the light path of the spec so that you could shoot a laser through it and measure the pH. Another challenge with analyzing anything in pore water in general is that you generally have very little sample to work with. So you can take like a 50 mil Falcon tube and fill it to the top with mud from the mudflat, and then centrifuge it, and sometimes you end up with less than 10 mils of water. And that's all you have to use to analyze the alkalinity or whatever it is you're measuring. But the beauty of spectrophotometric methods is that they're somewhat scalable. And so I was able to scale it down such that I can measure alkalinity with as little as 8 mils of sample. And I'm maintaining that very high precision, which is 1 micromole per kilogram seawater. And another thing that we had to do to scale it down was there's a part of the method where you have to aerate the sample using pure nitrogen to flush out all of the CO2 that you've generated in the titration. But how do you aerate that small of a sample while measuring the pH at the same time with the spec without the bubbles getting in the way of the measurement. And so what we did was develop a hook-shaped air diffuser to aerate the sample, and that's what that looks like. It shoots the bubbles up so that they don't fill up the cell and get in the way. All right, so on the topic of shell hash, Brian already spoke about this a little bit, but spreading shell hash on mudflats has been proposed as a method to counteract the influence of ocean acidification on pore water carbonate chemistry. And there have been a number of studies that have looked into this and done experiments both in the lab and the field. And as you can see in column M, they all have different results. Some of them found positive effects on pore water carbonate chemistry, but no effects on clam density or vice versa, and others found no effects whatsoever. And I think part of the reason for this is because every study used different sizes of shell crush and different densities of shell crush. Some of them only measured pH, which is a problem because you really want those two carbonate chemistry parameters so that you can contextualize the entire carbonate chemistry system. And so when we were planning the shell hash experiment of our own, the first hurdle was figuring out how to crush raw oyster shell. Turns out it's really hard, and what you really need is a machine called a hammer mill, which can cost north of $10,000, which was not in our budget. So DEI's facilities team sourced a machine which is like a miniature glass bottle crusher, which is effectively a miniature hammer mill, and they modified it in such a way that it's now a shell crushing machine, and this is what that looks like. So as you can see, it makes an awful racket and an awful mess, but you put shells in the top and crushed shells come out the bottom. You can then sieve the crushed shell to standardize the sizes to different sizes of shell crush. And if you're thinking, well, this is sounding like a factorial experiment, then you'd be right. So as Brian mentioned, we set up an experiment that looked like this using plots on a mudflat in South Portland containing different shell crush sizes and different shell crush densities, as well as predator deterrent netting and control plots that had no shell on them. This is what setting up the plots look like. If you look closely, you can see me and Sarah and Brian and Chad Coffin and a bunch of AmeriCorps interns helping out. All right, so then on the carbonate chemistry sampling side of things, we ended up sampling a total of 4 times. We sampled the day before we put any shell down to get sort of a background measurement of conditions on the flat. And then we sampled the day after putting shell down to see if there were any immediate effects of the shell hash. Then we came back a month later in May and sampled again. And then finally we came back in the fall in October and sampled all the plots with and without nets to determine whether any effect of shell hash on the carbonate chemistry lasted. All right, and these are the first results. So this is a plot of pore water pH across time in plots with different levels of shell hash. And so what we have here is I have grouped all of the measurements first by month. That's April, the day after we put the shell hash down, May and October. The red line is the median background pH measurement with the interquartile range. And then within each month, I have grouped the measurements by shell size. N means none, S is small, and then medium and large. And then within those groups, they're ordered from none to low to medium to high shell density. All right, and so this will give you an idea of how the pore water pH changed over time and in the different shell hash treatments. And what I found with statistical analysis was that the presence of any shell at all increased pore water pH. There was an ever so slight effect of shell size. So the smaller the shell crush, the higher the pH, which could be due to like dissolution dynamics. Smaller shell will dissolve faster than larger shell. We found no interaction between shell size and density, which is interesting. So it didn't matter whether there was a really low density of shell or whether there was a high density. Any amount of shell appeared to have the same effect on chemistry. And then finally, in terms of timing, the pH in April and May were pretty similar, and then the pH was quite a bit lower in October, which is probably a seasonal effect. It's colder in October, which means that CO2 is more soluble in water and the pH and omega are going to be lower. Okay, and now looking at omega, which again is the other really important carbonate chemistry parameter for the health of bivalves and other marine calcifiers. I have all of the measurements ordered in the same way. And you can see that omega appears to be a little bit higher in May, and there appears to be some influence of the shell hash if you compare the white box all the way to the left of each month, and then compare that to everything that comes after it. There appears to be a boost in omega relative to control, at least in May. Okay, so then what we found there was that unlike pore water pH, there was no blanket impact of shell hash on pore water omega. The shell size really appeared to be the more important factor here. So the smaller the shell, the higher the omega. And omega was higher in May and lower in October, just like pH. So in summary, we successfully manipulated mudflat pore water carbonate chemistry, meaning pH and omega, using shell hash. However, as Brian mentioned, we observed too few clams to determine whether shell hash improved their abundance. We can't even run the analysis because we just have too few observations of clams. And then what I'm wondering is, like present day conditions on this mudflat are not nearly as bad as we would expect them to be in 30 years, in 50 years, and so on. So I'd really like to do a follow-up project in which we maybe do an experiment in the lab to determine whether shell hash is more effective at buffering carbonate chemistry and improving clam abundance under future pH and omega conditions. All right, so we made the front page of the Portland Press Herald. That's Sarah putting down some shell hash in a plot. So that was really cool. And I just want to put a plug in for my own presentation next week, where I'll be telling you about the results of my carbonate chemistry monitoring project on 2 local mudflats and peels. So that one is not about manipulating the carbonate chemistry, it's about observing the carbonate chemistry and how it changes throughout the entire clam growing season. So with that, I'll open it up to questions for me and Brian.
Thank you, Rob. So if anyone has any questions, raise your hand or write it. You can also write it in the chat. We covered a lot of information today.
Yeah, thanks, Rob, Brian, this is Daphne in the conference room. Really cool sets of experiments. A couple questions. So one, when you went back after 6 months, did you see was the shell hash like still there? And did you track any of that? Like how well it held up?
Yep. So the shell hash was mostly still there in October. I think probably a little bit of it in the plots got washed away, but more so what we saw, and especially when me and my team were sampling mud samples for alkalinity analysis, when we dug into the mud with a spatula, we found more of the shell hash kind of buried under an upper layer of sediment.
Mm-hmm.
And that was kind of the effect that we were hoping to see, because we wanted it to become incorporated in the sediment where it would presumably have a greater effect over the long term because it's continuously dissolving.
That's a great question, Daphne, thanks. I'll just add a few things, which was when we started talking about the design, we actually talked a lot about whether or not we should be digging the shell in, or whether or not we should just simply be spreading it on the surface. And we decided to spread it on the surface because what we're also trying to do here is some kind of application that communities or individuals might use. And we didn't think that people would take the time to rake in shells into a mudflat. Just if they had the chance to do it, they probably would not opt that way. And so that's what we did as well. One of the things that we did, however, that I think did help is that when we first put those shells down, we actually took watering cans and watered the plots so that as the tide came in, the top chips might have gotten dry and would have been lifted by the water. And so we did see some of that, and that's how shells might drift out of the plots. But if they're wet, they did just fine. So we were pleased to see in October that underneath that small, if you will, that duff layer of new, sediment, the sediment that, if you will, landed in our recruitment boxes, covered up most of that shell, so it kept it in place.
Mm.
Yeah, and that's a concern that also sort of influenced our selection of shell crush sizes to a certain extent. So we could have gone even smaller and put down like powdered shell, which, for all we know, that could have had an even greater effect on the carbonate chemistry. However, we had reason to believe that it would all just wash away in the tide.
Yeah.
So we still used a small shell size, but we went a little larger than powdered shell.
We just wrapped up an experiment on a clam farm in New Jersey where we were collaborating with a farmer, and I appreciate very much how careful you guys were with the size of shell fragments and planting densities and those kinds of things. In our case, we left it up to the farmer to go and put the shell out, and he did that. Alex knows this because she's been out on those plots. He went out with a barge that's about the size of this room and just blew shell overboard, and it ended up being probably 2 or 3 inches thick at least. And it was big gnarly chunks of clamshell, and it worked really, really quite well. And we actually seeded over top of the shell. We let it sit there for about a month and a half so that it got silted in a little bit, and then we put hard clam seed down on top of that, and it actually performed better than contemporary nets did at his site. And so, I wonder sometimes if, there's a go big or go home aspect, but I've also seen the opposite where there's been, if we put too much shell down, it goes the opposite way and doesn't do what we think it wants to do. So I just think I'm surprised at your biological result. I think I would have expected to see clam recruitment, especially given Mark Green's results in that same location. So confusing result.
Well, it is confusing in a way, but it also is not confusing in that I don't believe Mark has data on, green crab abundance. But all we've seen is an increase in green crab abundance through time. And so his study was done, I think, either 2010 or 2011, and ours was 2021. My guess is that there were more green crabs on that same flat in 2021 than there were when Green did his work. And so that may have influenced differences that we saw.
Yeah.
We have a question from the chat regarding lower omega and pH in October rather than spring. Is the temperature really that much lower in October than April that would lead to those changes in carbonate chemistry parameters? Do you think instead it could be net ecosystem metabolism effect, which means less photosynthesis and more respiration in October than April?
So Ray brings up a good point, and I kind of glossed over this in my presentation. So there's a bit of a temperature effect seasonally, but also we have the effect of primary producers, meaning phytoplankton, bloom in the spring and proliferate throughout the first half of the summer. And that brings pH and omega up because they're photosynthesizing and using up the CO2 that's in the water. But then in the late summer and early fall, those blooms die off again, and then all the carbon that they've collected gets broken down by microbes, and that reduces the pH and the omega again. So yeah, there's definitely an effect of that there, and that's a big part of why we're seeing those seasonal differences in the figures.
All right, another question from the chat. Is there much heterogeneity in the pore water chemistry within a flat, vertically or otherwise, or do flats tend to be pretty well mixed?
Yeah, so there is a pretty dramatic influence of sediment depth on sediment chemistry. I assume that's what you mean by vertically. So what we did for this study and in my monitoring study was standardize the depth to which we were sampling. And I went with the upper ½ centimeter of sediment. The reason being is because that's where settling juvenile clams are going to be.
I had a question. So as a person that works in the mudflats, I'm aware that there are, it's not like they're barren of shells, like there's a lot of remains of shells that are in the mudflats, some deeper than others. And what effect do you think, do you think that those are having effects on anything? I was just wondering your opinion, Rob and Brian.
To shell pieces that already exist in the mudflat influence carbonate chemistry. Yeah, I think so, absolutely. But that's that's part of the natural order of things, and there isn't enough of it in there to buffer the pH change that we would expect to see from future ocean acidification. We put a lot of shell down in these plots, and it took that much to see even marginal effects on pH.
Any other questions from anyone? Colleen, our education director, asks, is it possible that allowing more time would show differences in both of your sets of data?
So in terms of carbonate chemistry, any effect that we had seen earlier in the spring pretty much appeared to have disappeared by October. And so I don't think allowing more time would help there. What I wish we could have done is sample more in between May and October to really target the moment in time that the effect began to disappear. And also determine at what stage settling clams might be at the same time.
And on my end, I think that if we had lengthened it out from, let's say, April to October versus April to December, I think we would have found even fewer clams in the plots because more crabs would have more time to have devoured the small clams. So I don't think we would have found as many clams as we did in the first place if we'd let it go for much longer. It's a good question. And I'd like to thank everybody for being here today. Thank you.
Yeah, thank you for a great discussion. And just as a reminder, next, as Rob mentioned, next week, we're going to be talking more about ocean acidification and effects in pore water on commercially important bivalves. And we're going to be doing it the same time, same date, same day of the week. So join us next Tuesday if you want. And then I put the full schedule up of the remaining seminars in the chat too. So please contact me if you would like to receive the Zoom link to those. And thank you everyone for attending.